pretty sure the answer is yes, but we're gonna treat these as far as the final exam goes. The presentations all will kind of work the same way as all the paper presentations throughout the term. Yeah. Okay.
Right, so there's going to be questions from them.
Yeah.
Yeah, yep, for sure. So, anything else? So, are you ready, Peter? Okay. Okay, so, oh. Sorry, laser pointer. And then to advance your slides, push that button. Go, push that. Don't push that back button because that's a black screen. Okay? Okay, all yours. Okay, Peter.
Um, so today we'll be looking at one of my work that I'm currently doing, and actually I started this during last summer, but being in this class, I thought it would be nice to just share it, and yeah, and this is part of why I'm in this class, to improve myself on the biological aspect of this project. So the topic here is using cyanobacteria for long-term sustainability of Martian colony through application of novel bioreactors. So the question will always be like, why do you want Martian colony? Like, why? Why can't we just stay on Earth and be cool? It's a monster and stuff. So let's look at it. So it is believed that the atmosphere of the Earth contains CO2 billions of years ago, and with algae bloom about 6 billion years ago. Um, we know when algae bloom occurs, the release of oxygen. So scientists believe that we came into existence through these cyanobacteria and algae. So algae and cyanobacteria were the first forms of life. That's what is believed. And one of the challenges for the colony of Mars is transportation of water, food, energy from Earth to Mars. This project is actually related to what NASA is doing, and is being partly funded by NASA also. So one of the challenges they encounter is how to transport water, food, and energy from that. And that is what this proposal seeks to address. So we have logistical constraint for mission design. That's for astronauts going to the space. Then the need for self-sustaining system to recycle nutrients, perform in situ extraction, and create oxygen and protein. So this simply means that on Mars we have the people there or the astronauts should be able to um, recycle the waste product they are using so that they can keep sustaining themselves. So just like you have wastewater, and instead of just sending out the wastewater, cyanobacteria can grow with the nutrients of this wastewater. So it's been recycled for them to have the water back and make food from this growth of the cyanobacteria and other useful components. So for this proposal, we'll be using bio-generative life support systems such as bioreactors, which can grow phototropes like cyanobacteria. And one of the critical issues is payload. How do you carry this water, this food, so that the spaceship is not weighed down, there is no excess weight. So that's one consideration also. Then we want to look at validation of this reactor, how cyanobacteria can grow. And once cyanobacteria can grow, that means the mass can support life. A lot of research has been done and even found out that cyanobacteria can grow on Mars, but this is looking at growing cyanobacteria and reducing the constraint of logistics in terms of weight of all these materials to the market. So why do we want to colonize Mars? I'll just run down because I made this slide so that it will be explanatory to everyone. So why do we want to colonize Mars? So curiosity. We are so curious and we want to move further, we want to expand. So we look at the economy, we look at expansion. Okay, if you talk about the economy, if you talk about the political dominance, if America is able to sustain life on Mars, it remain what power? So it's political driven and also economically driven because we improve the quality of life on planet Earth as we will not be having the opportunity to destroy the Earth as we've done so far. So the background, I won't be talking much on all this so that I don't spend much time on one slide. So, like I've said in the introduction, so food and water using and wastewater treatment to be done on site. So this is so much different between this background. This is just more insight on what we are looking at. So, let me move on to now. So, like we've done in previous class, we look at extreme environments. The masses can be said to be an example that has a very extreme environmental condition. So, what organisms can survive there? So, the extremophiles can survive there. So, if you look at this page, you can see that the mass is acidosis soil, low temperature, lack water, low gravity. So, these are the challenges on Mars if you want to grow an organism. You want to look into the conditions, how you can, which organism you can grow, and that's why we're choosing cyanobacteria. Although some scientists, like whom I showed you from the University of Kansas, they did on some archaea, like methanogens. Methanogens are still part of the family of bacteria, so those ones are there, but this one I'm looking at, the cyanobacteria, we can also refer to as the blue gene algae. So if we look at the temperature on Mars, you see that it's about minus 150 to 30 degrees Celsius, but Earth we have minus 88 to 55 degrees Celsius. So if we look at I shouldn't dwell much on this, but interestingly, what I will be looking at is the composition of the atmosphere, which is this, sorry, that, yeah. So the composition of the atmosphere, because that's where we want to be able to grow the culture, so which is the cyanobacteria. So for us to grow it, we are really concerned about the composition of the atmosphere on Mars. So I will still come back to that. So why cyanobacteria? I've talked about that. You know much more better than me. So these are still reasons because they are extremophiles. So extremophiles, we are looking at the pH, we are looking at salinity, contaminants, we are looking at the humidity and all that. So this is why cyanobacteria is good. And aside that, we have the social impact of cyanobacteria, creation of jobs. That's what you see here. So improve health, wastewater remediation, and a lot of stuff that cyanobacteria can do, and algae also. Sometimes algae is mistaken like it's cyanobacteria, but one is prokaryotic and one is eukaryotic, which is because of the cell in the nucleus. So now this is still economical algae, which is looking at the prokaryotic, the cyanobacteria. So if you look at it, you see that over this chart, the protein B compared to soya beans called ethanol, you see where algae is here at 50, and furthermore you can see algae that's growing up over time at sorry. So algae, yeah, 50, yeah, 20, comparing to these other plants. So then you see algae here. So also you see the growth over 60 years. So algae increased like 40 times, while soya beans 3,000 3 times. So you see the protein content of algae is much more than all these other plants. So, so my developed hypothesis. So I'll be looking at cyanobacteria can extract biological fixed which I've talked about, and a lot of scientists have also looked at that also. So analysis of shingle life, these are group of pressurized landers who are provided data for completion of Martian solid rock, showing like what they can feed sediment from uh, regolith. So what is, if you want to know, like regolith is like you have this rock and you have particles on it which covers the rock. So these microorganisms can actually feed on some of these particles of the rock. So one interesting hypothesis we developed is this one using species of cyanobacteria we should, as we know, using a membrane system to allow free flow while exposed to CO2 gas will result in the enhancement of CO2 mass transfer by the formation of inorganic carbon in the liquid. And so this was done by my PI, that's my supervisor in 2020. That's how I'm also working in mass transfer also. So the study objective is to validate the bioreactor system for cyanobacteria for possible development to support long-term sustainability of the Martian colony. So if successful, the work will validate the bioreactor system, offer significant reduced payload weight, increase protein capture, surface area. So to determine this, we need to create an environment that we can grow this. So we are looking at 1/10 of the atmosphere, and you know that the atmospheric pressure of the Mars is one over 25 of the Earth, which is about 0.06 psi or 6 millibars. So, and the Earth is like 14.7 psi. So we are looking at 1/10 of the atmospheric pressure. That's like 1.47 psi. Then we're looking at 95% CO2, 3% nitrogen, and 2% alcohol. So this is the environment we want to create for the algae. And now, a methodology. I know this is a biological, environmental biology, but as a mechanical engineer, this is my first work I'm doing currently. We need to design this system, which is a photobioreactor. Now, this has been designed. So this was used for for a wastewater treatment plant. Already, this has been tested, but now we are working on using the same mechanism here to test this. Why we can't use this is because of leaks, too much leaks here. So we need to be sucking pressure and it's not working out. So we need vacuum pump to suck the pressure out and the air out. So but it's not working. So we have to design uh, a two-chamber system where we have base gas supply of CO2. We have a carbon analyzer system. We have this vacuum chamber. So this vacuum chamber, we model everything. So we have the flow meter talking about the temperature probe, looking at the pressure, and looking at how to continue to remove air from the system. So that's why we are using the vacuum chamber, and inside we have the substrate where the algae is going to stay to grow. So let me just show you. So far, this is what we achieved during the summer, but this fall we've not been able to do much because of coursework. So over the summer, we worked on this just just like this design. This is what we drew to actually come out with. So far, we are looking at one 1/10 of an atmosphere. So what we have achieved now is just about 2 psi. So we are looking to go down a bit because we've not been able to achieve 1/10 of an atmosphere. Remember, this is the actual figure of the atmospheric pressure of Mars. So we've not been able to achieve that at this point. Now we've been able to put in feedback control system to check on Arduino on how to monitor the temperature, the pressure, and everything. And yeah, it's interesting and it's going well. So, so what are the challenges we're facing? So we look at solar radiation, dust removal, dust contaminant, and water requirement, and all these calculations are being done to give us a 1.35 liter per minute. That's the flow rate that will be needed to grow the algae. So if I dwell on this is something interesting that will waste much time. And so if you look at so, so we look at mass transfer, harvesting energy, payload reduction, and so one of the things I did put here is contaminant also. So one mistake is what we call it because when we launch a spacecraft, they didn't sterilize the spacecraft. They just launch it to Mars and up to space anyway. So they did not sterilize it because it's required that you sterilize the system before you send it to space because of contamination. So these are also some of the challenges we face. Considering. Now, the cyanobacteria culture, which is the main microorganism we want to grow to use. So we'll be using inorganic and polymeric materials to supply the material that can be without chance of contamination. Yeah. So the culture will be monitored in the 10-day period. We monitor the cyanobacteria formation through the substrate general work. We're using chemiluminescent method. This method is why we're using it because of the food. When this chemical reaction occurs, there is such a light that comes up in the reaction, so we produce photon. Cyanobacteria needs sunlight for kind of solar energy to grow. So that helps a lot. So we use pictures of substrate, yes, and our camera taking images that we grow while monitoring, then to ensure the nature of usual optical access to inside of it. So what this means is they let me quickly go back and show you. So if you check the material we're using here, it's transparent. This is to allow light to go in. That's what that part is saying. Then the expected outcomes is that the proposed work will facilitate the applications of humans on Mars by delivering oxygen from photosynthesis, recycle wastewater, and most importantly, produce proteins from nitrogen filtration or from in situ resources on Mars. That's from the entire um, wastewater from what they are using on the spaceship. Now, the lightweight, low pressure valve is designed to minimize payload compared with others. This is talking about the materials we use as well. So by growing the cyanobacteria continuous valve, the amount of water needed for continuous growth is 80% with the mixes. What we think we'll get now, and but we are looking at 90%. That's what we are looking at. The energy and time needed for harvesting the biomass is reduced by deploying a simple script in the day. So you want to know how to harvest this so we are still working on this. Cyanobacteria cultures will also be used to support the of course we know this. Then the final project, which is cyanocamine, that's about friendly material with many really powerful to power stack. So the proposed timeline is for about 9 months, but like I said, we started in summer, but we've not been able to move further. So at this point, let's say we spend like 2 months already on the project. And that's how it's going to go. Thank you.
Time for a few questions. So, questions from Peter.
You mentioned that you want to go down in atmosphere. When you say go down, do you mean like from 110 to 120?
Okay,
I think
let me
that's
go back to this. Yeah, so the atmospheric pressure of Mars is 1 over 25 of Earth. And we are not looking at this. We are looking at 110. I shouldn't have put this here, should be 110. So we are not looking at this, we are looking at 110 of an atmosphere. What is 110 of an atmosphere? We have the atmospheric pressure on Earth is 14.7 psi. So we are looking at 1.47 psi for the design, which is this design. So far we'll be able to bring this guy, this is a rotor meter, this is pressure guy, and we'll be able to bring it to 2.0 psi. So we want to take it down, and if we can take it below 1.47, it will be fine. Okay, I didn't put a system. We started with a vacuum chamber initially with a very light material, and when we drew it down to about 4 psi, it's like exploded. You know, not that much, but due to leaks, it just shattered. So that's why we increased on the material.
So, I had a couple of questions. So one of the things you stated is that Martian soil is toxic. How is Martian soil toxic?
Yeah. So I haven't really gone in depth. So like I have to consult materials. So in order for me to understand why we are using cyanic material and how this is extreme environment, I looked at the original material. So it's in toxic soil. So I cannot analyze it the way they analyze the chemical. So but they so there are some experiments that were done on some rocks or some rocks anyway, and they found out that um, the composition of these rocks can cannot support all organisms. So when I say toxic soil, it doesn't mean that every organism or every microorganism is going to just die because it's toxic. So it doesn't support most life. So I think that's what we
There's
use.
a big difference between something being toxic and not being toxic. So we need to go ahead and actually, the way the soil is toxic, is we found high levels of chlorate in the soil. So, let's see, another thing too, so the role of nitrogen, do you think that providing the 3% of nitrogen is going to be enough for nitrogen fixation, provide enough of that particular element of growth?
Yeah, like what we need to do. This is what we have in mass, 1.89%. So and we have like 70% of Earth. So we are looking at if we take 3%, what will come out. We haven't done that yet. So if we try it and works, fine. If it doesn't work, then we have to adjust the values. So it's not like
Okay,
we
so let's go ahead and I'm playing devil's advocate here. So let's say that the 3% doesn't work. Now you've mentioned other things that you're going to be utilizing. What would be another good source of nitrogen for these bacteria proposing to possibly use?
when you say source of nitrogen, and so if we are looking at wastewater, for instance, then we can get nitrogen from the so we can use the wastewater.
And so we'll address that in your proposal. Okay. And then, one of the things about this too, again, what I would recommend you doing is, essentially taking what you thought for your research project itself. But then for this project for class, one of the things I think you really have to be aware of is sources of light and all. And so, when you consider rovers upon Mars, the Curiosity and Spirit relied on the photocells, but it was very limiting because of the amount of light moving from the sun all the way to Mars, as well as dust storms and stuff like that. But I want to think about how essentially the photons that are required will need to be supplied, and not just through like the plastic of your current container, but sort of think about if you were to have this actually operating on Mars, what you need to do to ensure that there's enough light.
So, okay, that's like previously, discussing this program, so I asked about light, because I know, to show that it's dark over there in Mars. So it said, just worry about your design, when we get to that stage, fix it. But while I was doing my study, I saw the method like being used, which is chemiluminescent method. So these chemicals can actually help to that the link of light, which over time as they react. So I don't know if that's what, but I can see what you say out of what you should more research. What?
So why did you choose a plug flow reactor?
A what?
A plug flow reactor, why did you choose that?
A photobioreactor?
Yeah, why did you choose the plug flow? So it's named plug flow.
Plug flow. Is there this one? Sorry?
Only if I'm a plug flow reactor. Imagine if you regular plug flow reactor.
No, I mean, we are using a photobioreactor. This, yeah?
Yeah.
So this is a photobioreactor. Yeah, so we are looking at light, photo, right? Photon thing. That's why I talked about the transparency of this system and the Apex glass we are using because it's transparent. So why we are using this is because of, if you see this vertical chamber, it's able to retain the cyanobacteria on this membrane or substrate. So and it's easy to also harvest this or to scrape it, unlike just growing it on the
We're
wall.
able to do this, but I want to make sure that Abdul has enough time for his presentation.
Yeah.
And so, so I'm sorry, we're gonna have to end it there, but thank you. Okay. And then use this laser pointer here. Advance. There you go. You're welcome.
Before I get going, the plug flow reactor basically means how the water flows, essentially more than the being a photobioreactor or not.
What?
A plug flow reactor essentially means how like the water flows more than anything, and how it mixes more than the actual reactor itself being a photobioreactor or not.
Okay.
Yeah.
But I didn't remember mentioning that. Thank you.
I think this one is full. Yeah. So I'm doing a project, an abandoned project that I kind of worked on before. It's basically sampling and characterizing bioaerosols in an indoor air quality conditions. And why I abandoned it is because I didn't have much of an experience in bioaerosols, and my advisor was like, "You also got your thesis to work on, so you might as well just start working on that." So the first thing that we'll talk about is a bit of a background. So when we're talking about indoor bioaerosols, is the things that essentially what you do in any closed conditions will provide. So if I'm talking right now, I'm producing particulate matters that have some sort of microorganisms binding to them. And a lot of these sources are, as you can see there, that's humans, plants. If you have mold in your walls, all these things that can aerosolize and then get into the aerosol. And what we'll do is basically go and test in conditions that have these, and then use some of our air sampling machines and techniques, and then take those samples and then use them in some of the techniques that we use and learn at this class. As also as a bit of another background, in the U.S., I believe it was the EPA looked at the U.S. citizens spend about more than 85% of their time indoors. So that's why this is an area of concern, especially depending on what can be as you can see, COVID in the last couple of years. This is how, when I say anything that you can do indoor can release these droplets. So as you can see here, there's the optimization there. When you read the droplets, either depending on their size, if they are of a PM100, they will fall faster than a PM2.5. So that's why we concern ourselves mostly with particles of very smaller sizes, I would say PM5 and lower, because these are the ones that have shown persist a lot more. And then we concern ourselves with PM2.5 because these are the ones that bypass your in like the body filter system also. And that's how, again, aerosols would then have bacteria or viruses bonded to them, and then someone else breathes them in, and you can say. When we look at sizes, this is why we concern ourselves. Like I said before, we have, you can say we have 4 different regions, and this is depending on the size of the particle, that's how far it gets. And we concern ourselves with 2.5 or less because PM matters 2.5 because it essentially goes all the way down to your lungs. And then this is just a little example. This is the SARS-CoV-2, but essentially how big it is, and then you can see even a particle of that size, how it can bind to it, and basically stay, because of its size, stay in the air for a long while. So the objective of this is essentially to investigate the natural decay, if there is any of these bioaerosols whenever they're binded to these particles, and then how does the size impact, how does the particle size impact these microorganisms at home? And then what is the impact of environmental conditions, so that we'd see like if it's too windy, if it's how the temperature affects it, and how relative humidity affects it. So our experimental method will be, so I say indoor conditions, but it is hard, it's very hard to test that. So what we did is essentially make mini indoor conditions where we'll generate some bioaerosols, we'll put them into this chamber, and in that chamber we'll essentially do our sampling, and then from there we'll take that and then we'll measure it and see what's in. So for our bioaerosol generation, we use the collision nebulizer. It's basically, it's essentially like a little chamber that has liquid in it at the bottom, and then you will introduce the aerosols that you're looking at, or the bioaerosols that you want to look at. No, I should say introduce the microorganisms that you want to look at, and this will aerosolize whatever is in there and let it out as bioaerosol. As you can see there, the bioaerosols that we looked at, people look at is pollen, fungi, bacteria, and viruses. These are all these things you can introduce into this nebulizer. For our experiment, what we looked at before was E. coli, essentially introducing E. coli into this, and then, as I said before, it's a mini indoor like technique, I guess. But we have this little chamber with a fan, with a mixer fan, and then this chamber is what we use for what we introduce our bioaerosols into. And then this type of filter here is just for to maintain the flow, more or less, than anything. It's not really the one that we use to look at what's inside, because you'll always, in a condition like this, you'll always have some loss, and you don't want the flow to be too fast or too slow. So this kind of like equalizes a bit of the flow. And then that goes into three things that we looked at. We looked at a gelatin filter, which is a normal gelatin filter. I'm pretty sure you guys have seen them before. And then we use an impactor. So what the impactor does is, so as you can see here, I'm just going to count it. It's like a lot of small cylinders, and all of those small cylinders have different sizes. So the particles will basically be getting caught depending on their size in that impactor. And it works similar to a cyclone a bit. It does have a little bit of a centrifugal force that kind of like knocks these aerosol, the particles in, let it go. And at the bottom here is, I can tell you now, is what we find to be the most efficient. I don't have this data on hand, but this is a bioair sampler. So this will essentially take the bioaerosols and do the job in almost immediately. We found this to be more efficient than, especially the gelatin filter. Gelatin filter had a lot of problems with the flow. I think even with the HEPA filter, it just couldn't really capture anything. Oh, no, really. So how we measured it was we used two methods. We used the culture method. So as you can see there, we take the results from our three filters, we transfer it into a broth. We take the broth, mix it, inoculate it, and then let it grow. And all of those are like the vortex mixed into the fuse. So that's what we did. And then the other thing we did was the Barisol. For the Barisol was the molecular method. So basically here, like we did before, you basically like separate the DNA, I believe, and then you would put it in like separate the DNA by breaking the cells, and then you extract the RNA you need, and then we use the qPCR, which I'm pretty sure for a while we didn't have. That's why it took so long for it as well. We're determined by the PCR, so and that's the process right there, lysis binding the Washington relation, which we learned as well. So we use the qPCR to determine the number of viable and non-viable bacteria or virus that was that came in, because like I said before, one of the challenges of doing it like this is how the flow affects the bacteria, because a lot of the things don't get caught, and that's why I said the bioair sample was the best, because of it being designed to work like that. It is designed to take in flow, essentially, and then spit out something that you need. And then, again, these are things that we learned, heating and cooling it, and nature, and the immune cycle, and then detected by the by basically conventional fluorescent signal is what we use for that. And then, so from there, for my actual experiment, this is where we stopped. We couldn't get past this because, again, I did not have the time to look at it at all. And also, we didn't have, since we were in the environmental engineering department, we didn't have any of the easy access to the UW machines, because even if my advisor emails someone in like the chemistry building or like the biology building, like the bio building, they're like, no, we're using it.
Should have come talk to me.
Well, it was based on him, so it was my choice. Now that I know you.
There you go.
So from there, this is what I expect some of our results will be. So then I did a little bit of research on how experiments like this have been done on some different indoor air conditions. And as you can see here, that's the virus concentration. And this is depending essentially on their size. And these 3 out of 9 means they essentially had 9 samples from this one. They had 4 samples from here, and then 3 samples from these different environments. And then that's what they found in the. So then 3 out of 9 means they found the viruses in 3 out of those 9 samples. And then the colors essentially denote the concentration of those viruses in that particular sample. So as you can see, and again, even in the health center, you see a lot of it is in the very small size range here, but the concentrations aren't very high. The bigger problems we have, like we saw with COVID, is stuff like daycares and schools. As you can see, there is a very high concentration of comparatively to with the size and the concentration of the viruses. The thing is, even when I say that, even with the size, even if it was higher than that, it is still not good for you. Even if you apply the filters, it doesn't mean that you should just go ahead and stick your face in there. and then for comparison, they did it also in an airplane, which they still found 2 out of the 3 samples in there. They still found it. I talked about how temperature affects it as well, as you can see, because I forgot to mention. This chamber here is also, I forgot to put this in the render here, but this chamber also has temperature and humidity controls to it. So we essentially did this experiment with differing temperature and humidity like control points to see what we asked. So this is some of the expected results as well. So as you can see here, this is at 20%, at 20 degrees Celsius, 25 degrees, 30, and 35. And then that's the concentration of the coliform forming units. Afterwards, I forget the
Colony forming units.
coliform units. But yeah, and then this is for the relative humidity at 50, 60, 70, and 80%. And then that's the concentration of the colony forming units as well. So this is so the thing is that, as I said, I don't really have results for this experiment, but I will more than likely expect, even in that closed setting, in that more closed usual setting, the results should mirror closely these results that have been done in actual indoor settings that are like a daycare. This was done in a chamber. This experiment here for the temperature and the relative temperature. But with that, if you have any questions.
In a very, just, I guess, layman's sense, the liquid impinger, does it work basically in reverse of a nebulizer? In a very basic sense.
Kind of yes and no, because it will kind of tell you what's in that bioaerosol more than, or like give you a. It's not gonna liquidize it if that's what you.
Okay.
Even if it's a liquid impinger, it essentially will. It will, however, give you something that then you can use in like.
I actually thought that
Or it does, it doesn't.
I thought it actually would have the inflow of the air and the materials in the air, and then the individual part is essentially water or some sort of buffer.
Okay, so actually it
And-
does.
and
So yeah, I
right,
understood it.
So
I
that's
understood it.
why like
It
everything
does.
would be caught
Okay.
in that liquid.
Because
So-
that's what then you essentially use straight to
Right.
to put it into your menu. Sorry about that.
Yeah. Okay, so again, I have to play devil's advocate because that's my role. So what sort of like new information are you trying to get from these studies?
The study is
So, because it seems like what you're trying to do is replicate some of the stuff that's been done before.
It is, the purpose is try to actually come up with an experiment that can be replicable and actually give you replicable results. Because even when I say expected results should somewhat match with that like post experiment, what we find is in a lot of these experiments, it's the conditions affected so much that even when you talk about natural decay, a lot of natural decay essentially happens when like a particle will fall on the ground. But in some of these conditions, a particle can stay in the air for a very long amount of time, depending on the size. So even like, so what we want to get from this experiment is an experiment that's replicable, and then an experiment that, and then there's something that they don't really look at is how, even when I say at the end they look at the size and how they categorize it, they don't look very much at the size impact of the viability of microorganisms. And what I mean by that is that how, depending on how small the microorganism is, can actually sustain